Physiologic pH, where pH stands for potential of hydrogen, is a way of quantifying the balance between acids and bases in the body. In fact, the pH depends on the concentration of hydrogen ions and can be described with this equation: pH equals negative log base 10 of the hydrogen ion concentration. Now, the cells and enzymes in our tissues and organs work best when the concentration of hydrogen ions is 40 times 10 to the minus 9 equivalents per liter, otherwise known as 40 nanoequivalents per liter. Small changes to that number matter a ton, and because it can get annoying working with such tiny numbers, scientists converted this concentration into a logarithmic function and expressed it as pH. In this case, a hydrogen ion concentration of 40 times 10 to the minus 9 equivalents per liter works out to a pH of about 7.4. With this logarithmic function, there are two important aspects to remember. First, as hydrogen concentrations increase, the pH decreases. And this is because of the negative sign in front of the log. Second, since it's a logarithmic function, pH and the hydrogen ion concentration don't have a linear relationship. For example, an increase in pH from 7.4 to 7.6 means a decrease in the hydrogen concentration of 15 nanoequivalents per liter. Taking the same change in pH but going the other way from 7.4 to 7.2 means an increase in the hydrogen concentration of 23 nanoequivalents per liter. This is why the graph of hydrogen ion concentration versus pH has a curve to it, rather than being a straight line. For simplicity's sake, when the body's pH drops below 7.4, it's considered acidemia, and when it goes above 7.4, it's considered alkalemia. So, due to this logarithmic relationship, a change in pH in the acidic range, or below 7.4, will show a larger change in hydrogen concentrations than if the same change happened in pH in the alkaline range, or pH above 7.4. Now, maintaining a pH between about 7.37 and 7.42 is essential for the human body. And this is accomplished with buffers. In everyday language, a buffer is something that acts like a protective cushion or shield. And the same is true of physiologic buffers. They shield or prevent the pH from rising or falling too fast. The reason the body needs buffers is that acids, or molecules that readily give up their hydrogen ion, are being generated by the body all the time. These extra hydrogen ions would shift the pH into the acidic zone, so the body needs a way to handle them without a major shift in the overall pH. To accomplish this, buffers can essentially take on some of these extra hydrogen ions, and therefore keep the pH from dropping too much. Buffers are usually a weak acid with its conjugate base, or a weak base with its conjugate acid. The most important buffer in the body is the weak acid carbonic acid, or H2CO3, and its conjugate base, bicarbonate ion, HCO3-. Carbonic acid forms when carbon dioxide combines with water, with the help of the enzyme carbonic anhydrase. As a weak acid, carbonic acid easily dissociates into hydrogen ions and bicarbonate ions. Of course, these reactions are reversible and can happen in the opposite direction as well. In fact, because carbonic acid is such a weak acid, when hydrogen ion concentrations get low, it'll drop off its hydrogen ion and the equation moves to the right, producing more bicarbonate and a hydrogen ion. And when there are lots of hydrogen ions around, the bicarbonate will bind to one and form carbonic acid, which can go the other way and split into water and carbon dioxide. At that point, the extra carbon dioxide can be breathed out through the lungs. Alright, so imagine you've got some extracellular fluid, and it has a normal amount of hydrogen ions in it, which puts it in the normal physiologic pH range. Now we drop some NaOH in, or sodium hydroxide, which is a strong base. That means that in water, it completely dissociates into sodium or Na plus ions and hydroxide or OH minus ions. The hydroxide ions bind to hydrogen ions and form water, which makes the fluid more basic, since there's a decrease in hydrogen ions, which increases the pH a lot. When there's plenty of carbon dioxide around, though, it reacts with the water to form more carbonic acid, which splits into bicarbonate ions and hydrogen ions, which quickly replaces the hydrogen ions, and therefore buffers the pH and keeps it in a normal range. On the flip side, imagine tossing HCl into the fluid, or hydrochloric acid, which this time is a strong acid. It would fully dissociate into hydrogen ions and a bunch of chloride ions. Without a buffer, this would cause it to become acidic, since there's more hydrogen ions, and therefore the pH would go down. The new hydrogen ions, though, get instantly picked up by bicarbonate ions and converted to carbonic acid, which would then dissociate into carbon dioxide and water. Once again, this buffer normalizes the pH. Now, the great thing here is that there's a huge supply of carbonic acid in the body, because it's formed from carbon dioxide and water, which are in abundant supply in the body. Finally, as a last point, if there's too much carbon dioxide, it gets blown off by the lungs. And if there are too many bicarbonate ions, then the kidneys eliminate them in the urine. Apart from the bicarbonate buffer system, there's also the phosphate buffer system. This is a mixture of the weak acid dihydrogen phosphate, or H2PO4-, and its conjugate base, monohydrogen phosphate, or HPO4-2-. Dihydrogen phosphate has two hydrogens, and it's always ready to give off one of them and get converted to monohydrogen phosphate, which has a single hydrogen. Now, the extracellular fluid, and especially the plasma, is also full of proteins, like albumin, which serve as another extremely important buffer system. This is because some of the protein's amino acids have exposed carboxyl groups, or COOH, that act as weak acids, meaning that they're ready to release hydrogen ions when the pH starts to rise. At the same time, other amino acids might have exposed NH2, or amine groups, that act as weak bases, meaning that they can bind a hydrogen ion and prevent it from decreasing the pH. In other words, a single protein molecule can function both as an acid as well as a base, depending on the pH it has to deal with. But unlike the bicarbonate buffering system, there are only a limited number of proteins in our plasma, which limits how much they can buffer. Okay, until now we've explored the extracellular fluid, but intracellular pH has to stay balanced as well, and this is the pH inside cells. All cells are full of proteins, and those proteins are the most significant intracellular buffer. An example is hemoglobin inside red blood cells. Hemoglobin can reversibly bind either hydrogen ions, which bind to the protein itself, or oxygen, which binds to the iron of the heme group. And the interesting thing is that when one of these is bound, the other is released. So when red blood cells are in the capillaries of various tissues, the oxygen levels are low and the carbon dioxide levels are high. In this case, carbon dioxide diffuses into red blood cells, where carbonic anhydrase enzyme combines water and carbon dioxide to form carbonic acid, which then dissociates to hydrogen ion and bicarbonate. Because there's so much carbon dioxide around, the reaction continues in the direction of generating more hydrogen ion and bicarbonate, and over time the buildup of hydrogen ions can cause the pH to fall. To prevent the hydrogen ion concentration from rising too quickly, each deoxygenated hemoglobin binds hydrogen ions. The bicarbonate, on the other hand, moves into the plasma in exchange for chloride ions. This exchange, called the chloride shift, keeps positive and negative charges balanced. Now, once the red blood cell gets into the capillaries of the lungs, there's a low level of carbon dioxide and a high level of oxygen, so the process reverses. Oxygen binds to hemoglobin and hydrogen ions get released. Bicarbonate re-enters the red blood cells and combines with hydrogen ions to form carbonic acid, which dissociates into water and carbon dioxide, and then the carbon dioxide gets breathed out by the lungs. Intracellular fluid also has large amounts of organic phosphates, like adenosine triphosphate, or ATP, and glucose 6-phosphate, which can both serve as intracellular buffers. The phosphate group of these organic molecules can serve as a source or a sink for excess hydrogen ions. Alright, as a quick recap. Between 7.37 and 7.42 is physiologic pH, and the body uses buffering systems to maintain pH within this range. Buffers are pairs of a weak acid and its conjugate base, or a weak base and its conjugate acid, and their physiologic role in our body is to resist pH changes. The most important buffers in the extracellular fluid are bicarbonate, phosphate, and plasma proteins. And the most important buffers within cells are proteins like hemoglobin and organic phosphates, like ATP.